Bismillah Rahman Rahim. Welcome again, guys. Now, welcome to another video lecture by Kami Biology. Today, in this video lecture, we are going to talk about cDNA library. Now, this is the video lecture which is regarded to genetic library, and this is the part of recombinant DNA technology. Now, in recombinant DNA technology, we talked about different expectation of the cloning. We talked about vectors, screening, selectable markers, and in genetic, and we also talked the genetic libraries. And usually in genetic library, we talk the genomic DNA library, that how we can get the genomic DNA library, how we make the genomic DNA library. Okay, we talked in detail. If you missed that video lecture, it is present into my channel. Okay, now today in this video lecture, we are going to talk about cDNA library. Okay, now for the understanding of cDNA library, you guys need to understand about the genomic DNA library. Okay, in the previous video lecture, if you guys remember, we talked that genetic library are that techniques or that method due to which we can make multiple copy of the genes in inside a host cell, and by using by using the techniques of recombinant DNA technology. Okay, this is called genetic libraries, because in libraries we have multiple collection of the books. Similarly, in this case, we have multiple copies of the genes stored in a host cell in a petri plates. Okay, now how we can prepare, or how we can doing the cDNA library, and what are the difference between genomic DNA library and cDNA library, we'll be talking in this video lecture. So stay tuned and continue the video. So what are the cDNA libraries? Let me write. Okay, cDNA library. Now again, this is the collection of complement DNA, or different kinds of the complement DNA inside the host cell. We call that complement DNA library. Now how we can prepare it, how we can make it. So for the preparation of the cDNA libraries, we use only eukaryotic cell. And in this case, we can also use the eukaryotic genomes as well. Now why we use eukaryotic cells, and why we don't use the prokaryotic cell? Because when we use prokaryotic cell, prokaryotic cell doesn't involve the splicing mechanism, and in cDNA, to make the cDNA libraries, we must or we should splice the whole genomes. Okay, splicing is that techniques in which the uncoding region inside the genes are removed out by using the techniques, and only the exon portion or the coding portion is remaining, and we get premature we get the mature RNA, and from the mature RNA we directly get the protein, pure protein. But in this prokaryotic case, we just clone the whole genes. We get the whole genes to clone, and we didn't want to clone only the exon portion. We code both portion. Okay, and from that way we get the premature RNA. Then this is the too much complicated process in the genomic library, as we are doing in prokaryotic cell. This is the too much complicated. But in eukaryotic cell, whenever you are prepare the cDNA library, it is too much simple as compared to the prokaryotic cell. Okay, now if we want to differentiate between them, so we can say that cDNA library is much more simple than genomic. So the second thing is that it is simple method as compared to genomic DNA library. Okay, now in this case, they can only express the coding region. Okay, now what actually we are going to do in this case? So in this case, again, we take the human cells, we treat it with the detergent, and after some time we get the whole human genomes again in this case. Okay, now this is the whole genomes. They contain exon as well as intron. Okay, now what actually we are going to do? We using the phenomena of the splicing. Now we put these genes into a eukaryotic cell. Now once we loaded the genes, it is any kind of the genes. It may be the human genes, bacterial genes, plant genes, any kind of genes it is. Now we should loaded it inside the eukaryotic cell. Now once we loaded here it inside the eukaryotic cell, then inside the eukaryotic cell, different machine, the genomes use different machinery of the eukaryotic cell, and after some time what actually happen, these genes having the intron portion are released out, and we only get the exon. So when we get the exons, so the genomes are now very simple. So the genomes are very simple. Why? Because this genome contain intron plus exon, and this genome as we get after the splicing, this contain only exon. Now exon is the coding region where the codon are present, and due to this way directly we get the protein, and intron are the uncoding region, okay, due to which we can't get the protein directly, we can get the protein indirectly. Okay, now how we get the proteins, how the introns are removed out. Now in this case we treat the whole genomes with the help of a protein we call that simple nucleotide riboprotein. So when we treat the whole genome with SNRP, simple nucleotide riboproteins, so then what actually happen, they can remove out the exon portion, and only the exon portion are remaining. So here only the exon portion are present. Let's say these are the exon portion. So to join each exon portion with each other and to make a mature mRNA, so what actually we are going to do? Now this is, let's suppose, the immature mRNA, or we can say DNA, let's say, and from the DNA we get the let's say this is the whole genome of the DNA. We get the RNA genomes, and after the RNA genomes we get the let's say this is the pre-mature mRNA or immature mRNA. And once we apply the SNRP, simple nucleotide riboproteins, so then they remove out the intron and only exon portion are remaining. So after that we add another structure, another organelles, we call that spliceosomes. So once we apply here the spliceosomes, then what actually the spliceosomes are doing here? The spliceosomes can join each exon portion with each other, and finally we get finally we get a mature mRNA having the exon portion or coding portion. Now let's say this is the mature mRNA having coding region or having exon. Once we get mature mRNA, then what actually we are going to do? Now we use this mRNA as a template to make DNA. So after that we get the DNA. And this is the DNA RNA hybrid. But all of you better know mRNA can directly produce the protein. But remember, in this case we don't need the protein directly, or in this case we don't get the proteins. Okay, what actually we are going to do? we must prepare the DNA because it is actually the cDNA libraries. We must collect the cDNA, a lot of cDNA in a huge amount inside a cell. Okay, so that's why we don't produce directly the protein. Now what actually we are going to do, we make the DNA. How we make the DNA? By using the mRNA as a template. Here we loaded an enzyme, and all of you better know that enzyme is called as reverse transcriptase. So here we loaded reverse transcriptase enzyme. Now once we loaded reverse transcriptase enzyme, so they used mRNA as a template and produced DNA, and finally we get DNA RNA hybrid. Now this is in unstable form. So what actually after that we do, we treat this hybrid with the RNase enzyme, RH enzyme. Okay, so RH or RNase. Now once we treat it with this enzyme, so they can remove out this mRNA, and only they just leave or just place. Now by using this DNA as a template, okay, now by using this DNA as a template, we add here the polymerase enzyme, DNA polymerase enzyme, and after some time we get another strand. Now this is a template. Okay, and after some time so we get another DNA strand which is complementary to that DNA. Okay, so after that we get a complementary DNA. Now this is a complementary DNA strand. Why? The DNA which is produced from this old DNA, which is using as a template, we get a new DNA. Now this new DNA, both DNA are complementary to each other, and they are making means they are connected with each other, and we get a double stranded DNA. Now this is a complementary double stranded DNA. Once we get the complementary double stranded, a lot of complementary double stranded DNA we get, we store it, then we call it cDNA library. So we get a large number of cDNA library. Now after some time, what actually we are, now this is the end of the cDNA library, that how we can prepare the cDNA library. Now after that, what actually we are going to do, again we treat this cDNA library with restriction enzymes, and once we treat it with restriction enzymes, then they can produce a compatible ends. So then what actually we are going to do, once we treat it with the restriction enzyme, we get the compatible end of this DNA, and then again we join it with the vector. So again we have the vector. Again in this case, let's say I take a plasmid as a vector, and here we are loaded our gene of interest. So this is, let's say, cDNA. We loaded here our cDNA, and this is the recombinant DNA vector. And all of you better know, again we loaded this DNA to the host cell. Host cell could be a bacterial cell or E. coli, or we can say yeast cell also, eukaryotic cell also. We loaded this vector inside the host cell. By using transformation process, we treat the cell with calcium chloride to uptake the recombinant DNA. This is what, this is the recombinant DNA vector, and after that once they receive it, so then what actually we are going to do, we allow the cell inside agar plate to grow in a large amount. Then we get a colony, and after some time we doing the screening process. Okay, now once these cells are grow in a large so once we get a large number of the cells, so still we don't know that which kind of the cells having our own target genes or cDNA genes, which cells have. So for that purpose we again doing the process of the screening to identify the exact cells that have carry our gene of interest. Okay, and after screening, then we make sure that each cell contain our gene of interest. We separate out our gene of interest, we take out our product from each gene of interest, and we run it on electrophoresis to we run it on electrophoresis, and we make sure that this is the protein, and then we use each protein for our purpose, for our different kind of purposes. Okay, so that's how we can use the genomic or cDNA library. So as I told you, it is simple as compared to genomic DNA library, because in genomic DNA library we can clone the huge amount of genes, which is too much difficult. Okay, and in this method we can use only those portion of the genes which contain the coding region. Okay, and moreover we can clone it, or we can use, in this case we use eukaryotic cell to make more cDNA copy by using eukaryotic cell. But in genomic DNA we can use prokaryotic cell. Moreover, they can express only the coding region, and genomic DNA library can express the coding region as well as uncoding region, or exon and intron. So that's in a sense about the cDNA libraries. I hope you understand about the cDNA libraries. But if still you have any kind of a question, you can write it into a comment. Inshallah, we'll give you answer as soon as possible. If you like the video, make sure to hit the like button, share the video, and subscribe the channel to get more interesting videos like that. Thank you so much.